All lab tables will be disinfected by spraying with Amphel or Lysol before and after any lab work is performed. Paper towels used for wiping all disinfectant can go into regular trash can. Hands should be washed before and especially after lab work. Paper towels used for drying your hands can go into regular trash can. No food or drinks are allowed in the lab. Although the bacteria with which we will be working are not considered pathogenic to healthy humans, always assume they are pathogenic or infectious and act accordingly. Never ingest or smell bacterial cultures. Report any spills, broken glasses, or accidents to the instructor immediately. No horseplay in the lab. Open gas jets should never be unattended. Never leave unattended fire. Never reach across the bench. Always pay attention to your surroundings. Holding chemicals in live cultures can be hazardous. Never hold 10 to 15 glass tubes in your hands. Glass slides and red biohazard bags are not permitted. Long hair must be tied back. Shoes must cover entire foot. No sandals or clogs are allowed. No bare shoulders. You can wear a shirt, sweater, apron, or anything on top to cover your shoulders. Eye protection must be worn at all times, like goggles, not glasses, and an apron or lab coat should be on at all times. Goggles provided by us are sterilized in a UV cabinet and must be returned to the UV cabinet after every lab. Students may not be allowed to participate in lab without proper attire and protective gear. Heat fixed and stained slides are not contaminated. Uncontaminated slides for disposal can go to cardboard box. You may not take prepared slides out of the lab to study at a different location. Wet mount slides have live organisms on them and must be put into a dish with disinfectant for disposal. Used pipettes go to round plastic jars on tables, and the packaging goes into regular trash can. Discarded petri dishes, swabs, microfuge tubes go into red biohazard bags on tables. Discarded tubes of media, but not broken ones, are collected in wire basket on a cart in the lab. Take the tapes used for labeling off the tubes for disposal before placing them on the cart. Any plain broken glass without bacteria, such as glass tubes or slides, may be put into the cardboard box. If you break glass tubes with bacteria, the area must be saturated with disinfectant. Paper towels used for cleaning bacteria spills is contaminated and must go into red biohazard bag. Immediately notify instructor. Broken glass must be swept up or scooped up and put into metal canister for sterilization. Never put any glass slides or tubes or anything sharp in the red-orange autoclave bags. If you or your group has more than one plate, stack them on top of each other. Wrap the stack with one piece of tape and write your name group. Place them in the bin for incubation on the side counter. Label the tube with tape around the tube just below the cap. Do not use marker or Sharpie to label directly on the tube. Combine tubes and test tubes for racks for incubation. Cultures used for inoculating lab exercises. They are found on the side counter and must be placed in the label rack and returned to the side counter before you leave. The test tube racks are color-coded for each table. Stain bottles are found by the sink. All staining must be done at a sink and chemical hood for special stains. Gloves will not be provided, but feel free to bring your own. Each student will be assigned a microscope and has a number space in a cabinet. Your desk number corresponds to the microscope number. You are responsible for taking care of cleaning your microscope and reporting any problems with it. Chem wipes used for cleaning lenses are discarded in the regular trash. Before putting the microscope back into the cabinet, always wipe off the lenses, set the objectives to 4x, always do general cleaning of stains on the microscope, and wrap cord properly. Each student has 7 items in the drawer: slide box, inoculated needle, inoculated loop, marker, clothespin, ruler, and a lab apron. Claim one of the slide boxes for the semester and label your name, instructor initials, and the days you go to class. Slides and slide boxes may not be taken out from the lab under any circumstances. There are no open labs. You must finish your experiment during your lab period. You may not join another lab for extra time.